In the last class we have discussed about the general properties of the receptors. अलग अलग रिसेप्टर के बारे में आगे बात करेंगे जब हम सेंसेशंस की बात करेंगे, ठीक है, इसलिए उनकी अलग अलग हमने बात नहीं की। अब हम आते हैं सिनेप्स के ऊपर। सिनेप्स इज a site, as you can see, is a site of communication between the two neurons in the central nervous system. But this synapse, what I have spoken is only partly correct, is this one, between the two excitable cells. This is also partly correct because synapses has been seen between a neuron and a कौन सा सेल? नहीं, connective tissue cell of the brain, astrocytes are there. So communication can, and astrocytes are not excitable cells at least. But typically we define synapse is a site of communication between the two excitable tissues or two excitable cells. So what is an excitable cell? The basic question is, which cell can be called or labeled as an excitable cell? Any idea? We know muscle is an excitable cell, nerve is excitable, neurons are excitable, cardiac muscle is there, smooth muscles are there. Why the liver cell is not, kidney cell is not excitable? Okay, other connective tissue cells in the body, they are not excitable tissues. So what differentiates them from the non-excitable tissue? What is peculiarity that we call them as excitable tissue, others are not? Anybody? Any idea? Which cell is an, what determines the excitability of the cell? You see, every cell in the body has got a resting membrane potential. हर सेल में होता है, but the magnitude of that potential is differing in different cells. It may range from -5 millivolt interior to -95 interior negative. That is the range, broad range of the transmembrane potential in the body cells. लिवर के सेल में देखेंगे -5 मिलीवोल्ट ही हो सकता है. That is due to, फिर बातें शुरू कर दी, काटेंगे एक बार. That 5 millivolt is provided by either Donan's equilibrium effect, which is there, and partly because of the sodium potassium pump. Okay, but the other type of the membrane, other type of the receptors, channels are not there in other cells. So that is, so what I want to say is that any cell in the body which has got a resting membrane potential of -40 or more than that, -40, -41, -45, 50, 60, 90, अगर -40 से ट्रांसमेम्ब्रेन पोटेंशियल ज्यादा है तो वो सेल एक्साइटेबल होता है, वरना एक्साइटेबल नहीं होता. ये बेसिक चीज है. तो सिनेप्स is a communication between the two excitable tissues or two excitable cells in the body. There are two different types of synapses found: chemical synapses and the electrical synapses. In the chemical synapses, a chemical transmitter is being used by one cell to communicate with the another cell of the body or another excitable tissue of the body. Such where this communication is there, they are known as chemical types of synapses. These are the most abundant synapses in the central nervous system. सबसे ज्यादा यही मिलेंगे. इनके characteristic features अभी हम देखेंगे इसको. Then the second type are the electrical synapses. These electrical synapses are mostly found in the invertebrates also, and they have been evidenced in the human beings also. यहाँ भी होती हैं, but there are specific sites where they are found in most abundance. In cardiac muscle fibers, electrical synapses. In the smooth muscle fibers, electrical synapses are there. In the CNS, electrical synapses are there but only at the defined site. Otherwise, most abundant synapses in the CNS are of the chemical. These electrical synapses, we'll go in detail about these. They are via the gap junctions. Electrical synapses occurs where the gap junctions are there between two cell membranes where they unite. There is no gap between the two membranes. Gap is not there. Okay, they are known as gap junctions, but they are gap between because of the channels are there. They are named as the gap junctions. But there is no actual gap between the two membranes where the communication is taking place. And they are most found in the smooth muscle fiber or in the cardiac muscle fiber. Smooth muscle fibers दो तरह के होते हैं. Any idea? Functionally, two types of the smooth muscle fibers are there. Single unit muscle fibers and multi-unit muscle fibers. Single unit muscle fibers कौन से होते हैं? Which are single unit muscle fibers? Multi-unit means they are getting nerve supply. सब पे नर्व सप्लाई आ रही है और दूसरे वाले जो है सब पे नर्व सप्लाई नहीं आती. तो जिनमें नर्व सप्लाई नहीं आ रही, दे कम्युनिकेट वाया दी गैप जंक्शन. So electrical synapses, as I said, are found in the CNS, between the glial cells, between the neurons and the glial cells. And the main characteristic feature is that conduction of information via the gap junction is bidirectional. दोनों तरफ जा सकती हैं वो. A साइड से B में भी जाएंगी और B से A में भी जाती हैं. But chemical synapses are always unidirectional. One directional flow is there. Bidirectional flow is not possible in the chemical type of the synapses. So electrical synapses transfer the local responses in the faster way and their activity. जो electrical synapses हैं between the two and the excitable cells can be modulated, can be changed by the change of voltage in any of these cells. ये पहला सेल है, ये दूसरा सेल है. किसी में भी अगर वोल्टेज बदलाव इंडिविजुअली आता है तो इसको अफेक्ट कर सकता है वो ट्रांसमिशन को. चेंज इन द pH, concentration of the calcium ion as in the cardiac muscle fibers and G protein linked receptors, they all can modify the activity of the electrical synapses in the body or gap junction synapses. So these gap junctions के बारे में आपने पढ़ लिया होगा मेरे ख्याल से पहले. Wherever the gap junctions are there, अभी डायग्राम आएगा उसका. The two cell membranes oppose each other, पास में लगी हुई होती हैं, and at specific sites protein channels are there. These protein channels are known as connexon. ये connexon ही है जिनके थ्रू ट्रांसमिशन हो रहा है. And each connexon has got 6 subunits which form the channel for the flow of the ions, because that is the only mode available for the transfer of information from one cell to another cell. So these subunits of connexon are known as connexin, each with a molecular weight of 25,000. क्या यूनिट होती है इसकी 25,000 की? क्या डाल्टन्स होती है. बिल्कुल ठीक है. They conduct in both directions, conduct more rapidly in one direction than the other. This phenomena of unidirectional flow is known as rectification. ये हार्ट में पढ़ा था क्या? हार्ट में आया है क्या? यूनिडायरेक्शनल रेक्टिफिकेशन, एनोमलस रेक्टिफिकेशन का टर्म आया था. During plateau phase of the cardiac action potential, calcium going in, sodium going in via slow channels and potassium exiting out through the slow channels. Normally, most of the cell membranes in the body has got high permeability for potassium ion in both directions. But under circumstances, just like during the plateau phase, one directional permeability of the potassium is reduced. दोनों डायरेक्शन की उतनी नहीं रहती बराबर बराबर, एक तरफ की रिड्यूस हो जाती है. So that is a unidirectional reduction in the potassium conductance, and that is known as the anomalous rectification. Okay, and this rectification is also there in the gap junction synapses also. And channels can be open or closed, of course, depending upon the intracellular calcium or the hydrogen ion concentration. ये हम इलेक्ट्रिकल सिनेप्सेस की बात कर रहे हैं, ध्यान दीजिएगा. You can see this is the electrical synapse. There is no gap between the two membranes. ये एक membrane आ रही है, this is another membrane. These are the connexon molecules, and there is a tight junction between the cell membranes. The flow of information or the ions occurs through these connexons only. This is normal synapse, which is called as a chemical synapse, and there is a finite gap between this membrane and this membrane. So this is gap junction, electrical synapse. This is the chemical synapse. वही डायग्राम है और ज्यादा मैग्नीफाइड है. दिस इज कनेक्सॉन प्रोटीन. ये एक सेल है, ये दूसरा सेल है, और इसमें इंफॉर्मेशन ट्रांसफर हो रही है. दिस इज केमिकल सिनेप्स अगेन. सेम थिंग, मोर मैग्नीफाइड. Connexons are there, ions can flow across these channels only. ये आपका कनेक्सॉन के 6 सब यूनिट्स दिखा रखी हैं यहाँ पे. The flow occurs and there is no gap between the two cell membranes. Okay. Then comes the main part, जिसकी हम बाद में अभी बात करेंगे डिटेल में. Chemical synapses. Now chemical synapses in the central nervous system anatomically can be of 3 types. Exodendritic, most common. Axon of one neuron synapsing with the dendrite of the another neuron. Such a synapse is known as exodendritic synapse. Second is the exosomatic. Axon of one neuron and soma or cell body of another neuron. Such a synapse exosomatic. These exosomatic synapses, they are most effective in bringing out a change in the post न्यूरोनल सेल. जो इंफॉर्मेशन आ रही है सेल बॉडी के ऊपर, वो अगर सेल बॉडी पे आ रही है तो वो ज्यादा इफेक्टिव होगी बजाय डेंड्राइट पे आ रही है. बिकॉज एनी इंफॉर्मेशन रिलेड ऑन द डेंड्राइट will gradually decrease in magnitude as it reaches to the cell body and may not be able to generate an action potential at the axon hillock of the postsynaptic neuron. But if it is coming directly over this cell body, सोमा पे आ रही है, तो इसका इफेक्ट डेफिनेटली बहुत ज्यादा होगा. So exodendritic, exosomatic and exoaxonic. Axon of one neuron is communicating with axon of the another neuron. Such synapses are not rare, they are also abundant, but most the function of such synapses is the presynaptic inhibition. ये inhibition के काम आती है इस तरह की synapse. Exoaxonic synapses are working as inhibitors in the central nervous system. Mind it, CNS में excitation ही important नहीं है. If you don't inhibit certain pathways and certain neuronal circuits, the brain may go under chaos. Chaos create होता है, इसलिए inhibition बहुत जरूरी होता है. And exoaxonic synapses, most of them are playing or causing presynaptic inhibition, जिसकी हम आगे डिटेल में बात करेंगे. And these chemical synapses for transfer of information uses the chemical substance which is known as a neurotransmitter. So neurotransmitters are carriers of information from one neuron to another neuron. While some neurons along with the neurotransmitter also releases another chemical substance which is not a typical neurotransmitter but is capable of changing the effect of main neurotransmitter on the postsynaptic neuron. Such chemicals are known as neuromodulators. They are not typical neurotransmitters, but they are capable, they are released along with the neurotransmitter and they are capable of changing the effect of neurotransmitter on the postsynaptic cell. Such chemicals are known as neuromodulators. So a synapse then, which is a communication link between the two neurons, has got 3 features. A presynaptic terminal is there, a synaptic gap is there, finite gap is present between the two cell membranes, and a postsynaptic membrane is there. Presynaptic membrane is of that neuron which is bringing the information. Postsynaptic membrane is that which is receiving the information, and in between them is a finite gap which is also playing an important role in the transmission. This is presynaptic terminal, the postsynaptic membrane, and a finite gap is there. This nerve terminal contains most important what you call as the vacuoles are there, or synaptic vesicles are there containing the neurotransmitter. The most important content of the presynaptic terminal is the presence of the synaptic vesicles containing the neurotransmitter, plus they can have mitochondria also. The postsynaptic membrane is enlarged, it has got a greater surface area definitely coming for the communication, and again the most conspicuous thing for the postsynaptic membrane is the presence of receptors on its membrane for that particular neurotransmitter which is being released by the presynaptic membrane. So these are the receptors you can find. This is the postsynaptic membrane is there, synaptic vesicles are there, these are the neurotransmitters which are released. तो प्रीसिनेप्टिक टर्मिनल के नाम दिए हैं अलग अलग, ये सब उसके नाम है. टर्मिनल नॉब कह सकते हैं उसे, टर्मिनल बटन्स कह सकते हैं उसे, इनके शेप की वजह से नाम दिया हमने. एंड फीट भी कह सकते हैं, एंड फीट, एंड प्लेट. एंड फीट और एंड प्लेट में फर्क पता है क्या? यहां मैंने एंड फीट लिखा है, एंड प्लेट नहीं लिखा. एंड प्लेट कहां होता है? Motor end plate, that is postsynaptic. Motor end plate is a postsynaptic membrane. Okay, this is presynaptic part, and it can be known as a synaptic knob also. So multiple names are there of the presynaptic terminal. So what is the mechanism of transmission of information from presynaptic neuron to the postsynaptic neuron? That is the mechanism of synaptic transmission की बात कर रहे हैं. So arrival of the action potential, mind it, the information is coming via the axon, and axon are transmitting the electrical information in the form of the action potential. Most of them are transmitting by action potential. So action potential arrives at the nerve terminal, जिसको terminal button कह रहे हैं, वहां आ रही है ये पहली बात. This arrival causes opening up of the calcium channels which are voltage gated. ये सब important है. Each step is important because various drugs and chemicals are available to block the action to cause the anesthesia or other desired activity in the central nervous system while we are operating upon the patient or treating such this process. So हर step important because they can be interrupted and drugs are available and they can be generated also in the future. So calcium channels are opened up, and these channels are the voltage gated channels. It means the action potential which is coming down to the terminal lobe is causing a change in the membrane potential at that side, and that will cause opening of the calcium channels. This is linked because the degree of calcium entering, because entry of the calcium channel, opening of the calcium channel means that now the calcium will enter from extracellular compartment of the neurons to the intracellular compartment of the presynaptic terminals. वहां पे calcium घुसेगा, calcium की entry होगी. This entry of the calcium is voltage dependent. Mind it, action potential which is coming over the axon can have a full magnitude of 90 or 100 millivolts. It can have a magnitude of less than that also. It can be 80 millivolts also. It can be the 70 millivolts also, depending upon the situation. And that voltage of the action potential which is arriving at the presynaptic terminal will determine how much calcium is going to enter into the presynaptic terminal, because that is going to determine how much neurotransmitter is going to be released. That is going to determine how much postsynaptic cell will be excited. So all these things are interdependent of each other. So calcium channels are opened up. They are voltage gated channels. They are not other channels. Causes the entry of the calcium. Now calcium has entered into the presynaptic terminal. अभी हम पहले की बात करें. प्री-सिनेप्टिक टर्मिनल में आ गया. This calcium which has entered there combines with a protein present in the presynaptic terminal which we call the synaptobrevin and causes its phosphorylation. This synaptobrevin interacts with the syntaxin which is present in the presynaptic membrane. जो presynaptic membrane है उसमें प्रोटीन मौजूद है, उस प्रोटीन का नाम दिया हमने syntaxin. So the synaptobrevin then after getting phosphorylated interacts with the syntaxin because of the fusion of the vesicle with the membrane. So interaction is going on here, and ultimately this fusion of the vesicle with the membrane causes release of the neurotransmitter from the presynaptic terminal into the synaptic gap or synaptic gutter. So release of the transmitter is there, and as I have said, this release of the transmitter is related directly to the amount of the calcium which has entered by the change in the action potential or the change in the membrane potential of the presynaptic terminal. These are the events which are listed, thus I have narrated. ये तो गड़बड़ है थोड़ी स्लाइड. That is the whole process. अब हम इसकी बात करेंगे. So we have seen what happened at the presynaptic terminal, and neurotransmitter has been released. This neurotransmitter diffuses across the synaptic gap and reaches to the postsynaptic membrane. After reaching to the postsynaptic membrane, what this neurotransmitter will do or it can do, okay, it binds with the receptor present of the postsynaptic membrane. जो न्यूरोट्रांसमीटर रिलीज हुआ उसके लिए रिसेप्टर मौजूद है, वो मेम्ब्रेन पे भी हो सकते हैं और कहीं अंदर भी हो सकते हैं, उनकी बात करेंगे हम. So most of them are binding to the cell membrane receptors, and that binding of the neurotransmitter with the membrane receptors can cause multiple actions. Okay, so what actions can happen at the postsynaptic membrane in the neuron? Open ligand gated channels. Ligand का मतलब जो केमिकल वहां से रिलीज होके आ रहा है वेसिकल्स के द्वारा. Acetylcholine is a ligand, okay, adrenaline is a ligand, noradrenaline is a ligand. They are released there and they are uniting with the receptor present on the postsynaptic membrane. So these receptors which are binding, they are channels present there which we call them as the ligand gated channels. मतलब केमिकल यूनियन से चैनल खुली है ये, इसलिए ligand gated channels है. ये वोल्टेज गेटेड चैनल्स नहीं है, वोल्टेज बदलाव नहीं हुआ अभी, ठीक है, केमिकल ला के लगाए रिसेप्टर के ऊपर, उसकी वजह से चैनल खुल रही है. So they are ligand gated channels, and the property of these ligand gated channels is that they bring about a very rapid action. Action is rapid, action is short lasting, बहुत थोड़े ड्यूरेशन के लिए रहता है. ऐसे न्यूरोट्रांसमीटर जो ligand gated मैकेनिज्म से एक्ट करते हैं, उनका एक्शन हमेशा बहुत शॉर्ट ड्यूरेशन के लिए होता है. शॉर्ट ड्यूरेशन एक्शन रैपिड भी होता है, शॉर्ट ड्यूरेशन भी होता है, एंड ट्रांसमिशन इज वेरी फास्ट. द मोस्ट इंपॉर्टेंट एग्जांपल इज द ट्रांसमिशन कॉज्ड बाय द एसिटाइलकोलिन. एसिटाइलकोलिन के जहां भी रिलीज होती है एक्शन बहुत शॉर्ट लिव्ड होता है और बहुत फास्ट होता है, जबकि एड्रेनलिन का ऐसा नहीं होता, लंबा टाइम तक चलता है उसका इफेक्ट. द सेकेंड इफेक्ट विच दिस रिसेप्टर neurotransmitter binding can result into is activation of the G protein linked receptors. एक रिसेप्टर वो है जो चैनल से लिंक्ड है डायरेक्टली, दूसरे रिसेप्टर वो है जो जी प्रोटीन से लिंक्ड है, ठीक है. Then when the G receptor protein linked these receptors are activated, they can also bring about certain change. So the second mechanism of action of the neurotransmitter at the postsynaptic membrane is G protein linked receptor activation, and that can result into again multiple actions can be there. पहला वाला जो था उसमें सिर्फ एक ही एक्टिविटी हुई, आयन चैनल्स ओपन हो गई, ओके, एंड दैट ब्रिंग्स अबाउट द चेंज. दिस कैन कॉज ओपनिंग ऑफ द आयन चैनल्स, ये हो सकता है, मगर इसमें डिले हो गया थोड़ा एक्शन के अंदर. पहले जी प्रोटीन इन्वॉल्व होगा, फिर जाके आगे एक्टिविटी शुरू होगी. So ion channels can be opened. Secondly, cyclic AMP mechanisms can be activated. So opening formation of the cyclic AMP via activation of the adenyl cyclase mechanism. Adenyl cyclase is activated, acts upon the ATP, converts it into the 3,5 cyclic AMP, and then further actions are there. The third is activation of the intracellular enzymes can be there. That is the third mechanism of the G linked protein activation, and fourth activation of the gene transcription can be there. So such type of the activity require slow and longer duration action. इसमें लंबा समय लगता है इन सब में, ज्यादा देर तक एक्शन रहता है. So there are certain neurotransmitters which have got very short-lived action. Other neurotransmitter brings action which lasts for minutes, hours, and even days. Receptor activation is there. So multiple type of the hormones or receptors, neurotransmitters are there, bring about the action. So any of the above action can be caused by those neurotransmitters at the synapses. The action is always slower and long lasting. Whenever the G protein linked receptors are activated, receptor activation is slower and it is long lasting. So what this can result into? Neurotransmitter is released, there is a mechanism for that. They crosses the junction, reaches to the postsynaptic membrane, activates the mechanism. Okay, these mechanisms ultimately do what? Because if the postsynaptic membrane is to be activated, the information may need to be transmitted further down into the system, or it may end. What can happen is the electrical change has to take place in the postsynaptic membrane. That electrical change can occur either in the form of the depolarization, that is if the neurotransmitter and receptor combination can cause opening up of the channels, and the channels are say for the sodium and calcium, the postsynaptic membrane will be depolarized and the electrical change developed will be known as the EPSP, excitatory postsynaptic potential कहेंगे उसे. Okay, if receptor and neurotransmitter combination causes opening up of the sodium channel, closure of the potassium channel, or opening of the calcium channel, will cause partial depolarization of the postsynaptic membrane, and that electrical change is referred as the EPSP, excitatory postsynaptic potential. On the other hand, the other type of the activity which the neurotransmitter can cause on the postsynaptic membrane is development of the IPSP, inhibitory postsynaptic potential, that will occur if the potassium channels are activated. That will cause efflux of the potassium, cell will get hyperpolarized. जब भी पोटेशियम सेल से बाहर ज्यादा जाए, अंदर नेगेटिविटी क्रिएट करेगा एक्स्ट्रा, और वो हाइपरपोलराइजेशन कहलाता है. और it can be an opening up of the chloride channels which can move from outside to inside. क्लोराइड बाहर ज्यादा होता है, क्लोराइड अंदर कम होता है. अगर चैनल्स खुलती हैं तो क्लोराइड बाहर से अंदर जाएगा, अंदर की नेगेटिविटी को बढ़ाएगा और वो आपका आईपीएस भी क्रिएट करेगा. मगर इसमें एक पेच है क्लोराइड के साथ में. क्लोराइड के साथ में थोड़ा पेच क्या है कि इफ यू रिमेंबर द नस्ट पोटेंशियल फॉर द क्लोराइड आयन इज हाउ मच. डिफिकल्ट क्वेश्चन से, हालांकि ना किसका नहीं, आंसर यू आर नियरली करेक्ट टू द आंसर. द रेस्टिंग मेम्ब्रेन पोटेंशियल इन द नर्व फाइबर is minus 70 millivolts. The nest potential for the chloride is minus 70 millivolts. So if the channels are open क्लोराइड की चैनल बंद है, खोल भी दी जाएगी तो भी अंदर क्लोराइड जाएगा नहीं जब तक कि मेम्ब्रेन पोटेंशियल वही है। अगर अंदर का अगर मेम्ब्रेन पोटेंशियल घटा है किसी वजह से, ठीक है ना, तो फिर क्लोराइड अंदर जाकर के उसे इनहिबिट कर देगा, वापस 70 पे ले आएगा। तो सेल विल नॉट बी एक्साइटेड। So if the excitability of the cell is decreased, then it is also a kind of the inhibition. Then it is also a kind of inhibition. ठीक है? So hyperpolarization can be there. IPSP, inhibitory postsynaptic potential of the postsynaptic membrane. So these both EPSP and IPSP, they are not action potentials. At the site of communication, at the site of synapse, action potential is not generated. कहाँ बनेगा? एक्सोन हीलक के ऊपर, उस न्यूरॉन के पे। तो ये क्या है? IPSP, EPSP, local responses. Like the end plate potential in the skeletal muscle fibers, that is also a local response. This is also a local response. So at synapses in the CNS, only local responses are generated. No action potentials are there. So because they are local responses, they are conducted electrotonically. Movement to point conduction होता है, so conduction is always slower. पहली बात. Then it is a decremental conduction. It means as the conduction goes from this part where it is taking place, synapse is there, soma is here. The information travels from this dendrite to the soma. The magnitude of the response will decrease ultimately. यहाँ पे जहाँ एक्सोडेंड्राइटिक सिनेप्स थी, इस सिनेप्स से मान लीजिए 10 मिलीवोल्ट का बदलाव आया, सोमा तक पहुंचते पहुंचते हो सकता है 2 ही मिलीवोल्ट रह जाए. Decremental conduction is there. Action potential is all and none phenomena. It is conducted without decrement. So it is different from that action potential. It is a graded response. Graded response means more neurotransmitter, more will be the change, electrical change. Less neurotransmitter, less will be the change. Response is a graded response. It is not a all and none phenomena. नहीं है ये कि जितना होना है उतना ही होता नहीं है. It is always a graded response. And because it is a graded response, it can be summated. जुड़ सकती हैं ये. 2 EPSP जुड़ जाएंगे और एक EPSP आया है, दूसरा IPSP आया साथ में दूसरे न्यूरॉन से, तो ये घट जाएंगे आपस में. तो सोमा पे जो इंफॉर्मेशन पहुंचेगी वो घट जाएगी ऑटोमेटिकली. तो अल्टीमेटली द एक्साइटेबिलिटी ऑफ द पोस्टसिनेप्टिक न्यूरॉन is determined by all the input summation. Okay, so they can be summated, and of course they do not follow all and none law, and they can be both hyperpolarizing or depolarizing. That is EPSP भी हो सकता है और IPSP भी हो सकता है. These local responses again show something like cable properties. ये आपने पढ़ लिया है, पहले ही पढ़ लिया ना cable properties के बारे में. Length constant and time constant. Okay, ध्यान करो क्या होता है ये. Length constant is distance along which, ये पढ़ रहा हूं अभी मैं इसको, along the membrane at which the voltage signal is reduced to 37% of its original value. This is the dendrite. Synapse is taking place here. It has to travel to the soma of the postsynaptic neuron. Okay, here the maximum change was there and it gradually decreases in the strength as it passes. तो यहाँ से चलने के बाद में कितनी लेंथ ट्रेवल करी इसने कि उसकी ओरिजिनल वैल्यू का 37% ही रह गया. वो लेंथ कांस्टेंट है, हर न्यूरॉन का अलग अलग होता है. ये हर न्यूरॉन का अलग अलग होता है. लेंथ कांस्टेंट ज्यादा है तो इफेक्टिवनेस कम हो जाएगी, लेंथ कांस्टेंट कम है तो इफेक्टिवनेस बढ़ जाएगी. दैट इज लेंथ कांस्टेंट. देन टाइम कांस्टेंट इज देयर, अनदर फिनोमेना. अमाउंट ऑफ टाइम इट टेक्स टू रीच 63% ऑफ द मेम्ब्रेन स्टेडी स्टेट वोल्टेज. आपने स्टिमुलेट किया, मेम्ब्रेन पोटेंशियल में बदलाव आना शुरू हुआ. ओके, इट इज राइजिंग लाइक दिस एंड देन रीचिंग टू अ पीक एंड देन ग्रेजुअली. तो यहां से चढ़ने में 67% ये चेंज था आपका मान लो 10 मिलीवोल्ट का, और यहाँ पे 6.7 मिलीवोल्ट पहुंचने में कितना टाइम लगा, वो आपका टाइम कांस्टेंट है. टाइम टू रीच 67% of the maximum change is the time constant of a particular nerve fiber. So length constant and time constant are there, and they ultimately determine the effectiveness of the neuronal input at the synaptic junction. Summation or addition of information. As I said, EPSP and IPSP both can be summated together. Okay? And these summations are of two types. Temporal summation is there, spatial summation is there. In this CNS, this is more important than other places we have talked about. Temporal summation means this is a particular synapse coming from a neuron A to neuron B, and this neuron A is firing multiple. Information is coming via the same neuron at the same site only, at this single synapse only, or multiple synapses of the same neuron. एक न्यूरॉन ए 50 जगह सिनेप्स कर रहा है न्यूरॉन बी से, और वो न्यूरॉन ए लगातार फायर कर रहा है. ऑल दिस इंफॉर्मेशन विल बी समेटेड ऑन द पोस्ट सिनेप्टिक न्यूरॉन. दिस इज द टेम्पोरल समेशन. एक यूनिट से इंफॉर्मेशन पे जा रही है, लगातार आ रही है मल्टीपल टाइम्स में, दैट इज गेटिंग समेटेड, इट इज टेम्पोरल समेशन. दिस पोस्ट सिनेप्टिक न्यूरॉन is receiving information from hundreds of other neurons also. So those neurons, most of them may be excitatory. If they are falling on the one neuron, that will be again summated. Okay, that summation is known as the spatial type of the summation. So summation from a single input is temporal summation. Summation from multiple inputs on a single neuron is the spatial type of the summation.